Okay, so let's review. Naive B cells, on their surface, they express IgM and IgD. If a naive B cell encounters an antigen that matches its antigen binding site, the B cell becomes activated. And some of those B cells will, once that B cell undergoes mitosis, makes an army of cells. Some of those B cells will secrete antibodies immediately, and so that's secreting IgM. But these other B cells are going to undergo these processes known as affinity maturation and isotype switching. We covered isotype switching in the previous video, and now we're going to cover affinity maturation. And again, these processes go on at the same time. I've chosen to introduce isotype switching first. I'm now going to introduce affinity maturation. The purpose of affinity maturation, the function of it, is to improve the antigen binding site so that it has higher affinity for the antigen. You basically make antibodies that bind stronger to the antigen. High affinity antibodies are going to be much better than low affinity antibodies. How do we change the affinity of an antibody? How do we change the antigen binding site? It involves mutating it. So we're going to see mutations. All right. So let's say the B cell has become activated, and the B cell has gotten a signal from cytokines that came from helper T cells, and the B cell is told, you know what, we really need IgG to help fight in this infection. So we covered in the last video isotype switching, which involved enzymes AID and binding to the switch sequences, modifying the cytosines to uracils, and then the enzyme UNG modifying the uracils, and that will help you cut the DNA. This was isotype switching. And now we have constructed a heavy chain gene locus that uses the constant gamma region. So that's great. And that was isotype switching covered in the previous video. But now these two enzymes are also going to do something to the DNA region where you find VDJ gene sequences. They're not going to cut it, they're actually going to mutate it. So these two enzymes are also involved in affinity maturation. So let's see that in the next slide here. So I've just drawn the heavy chain gene locus a little bit differently. I drew the VDJ regions, and I've also drawn in the nucleotides in the junctions between them. So if you recall junctional diversity, there are the P nucleotides and the N nucleotides and the P nucleotides at the junctions. So this is this region of DNA is going to give rise to the variable region of the protein, where you find the antigen binding site. And then there's the constant gamma region, and that's going to give rise to the FC region of the heavy chain protein. So, we talked about B cell activation. When a B cell recognizes a antigen using its antigen binding sites in its surface Ig, then the B cell becomes activated. Now, again, remember how and why does a immunoglobulin bind a antigen? It has to do with the amino acids that are present in the variable region of the heavy chain and the light chain gene locus. And so those amino acids, right, where did they come from? They came from the genetic information present in the heavy chain and light chain variable region genes, right? So I just drew, for example, some DNA sequence that might arise due to VDJ recombination and junctional diversity, and obviously this is a very abbreviated version. But let's say this is the DNA sequence that is present in the VDJ region of the heavy chain gene, right? Due to the randomness of choosing a V, a D, and a J, and the junctional diversity. And this is what you get. And you got some amino acids here. Methionine, leucine, glycine, proline, I think that's asparagine. Anyway, so these amino acids give rise to the heavy chain variable region, and those are going to give this Ig some affinity for its antigen, right? So the shape that is created, the three-dimensional structure that's created by these amino acids in this order, gives some antigen binding site that seems to bind some molecule on the surface of the antigen, and that's great. But this could be better. We can improve on this. And how's that going to happen? It's going to happen by mutating this region of DNA and slightly altering the nucleotides and therefore amino acids that are present here. So the enzyme AID, which stands for activation-induced cytidine deaminase, so it modifies cytosines, right? It removes an amine group and it turns it into a uracil. So what the AID enzyme does in the variable region is it chooses a random cytosine nucleotide and modifies it. So I've chosen that one here, but really it could choose any one of them. So in every B cell that have become activated and is undergoing isotype switching, one cytosine is randomly chosen in the variable region. And what does this enzyme do? Well, it modifies cytosines and turns them into uracils. This is strange. Uracils should not be in DNA. Uracils should be in RNA. So the repair enzyme UNG, which stands for uracil DNA glycosylase, it says, hey, there's a uracil in this DNA. You got to get rid of that. Uracils don't belong there. So the UNG will go bind that uracil and remove it. Well, now we're missing a nucleotide. Well, repair enzymes are going to come in. What are they going to put in? Turns out they will try all nucleotides, right? So they could put in either a T or an A or a G, or maybe they randomly put in a C. So this is randomness, right? So we're mutating the DNA. It's really weird to think that when your B cells become activated, point mutations occur in the variable region of your heavy chain gene. This can also happen in the light chain gene. And so this process will result in possibly changing the codon that will code for the protein. So this whole process that we're doing right now that I'm demonstrating is known as somatic hypermutation. When B cells become activated, changes occur in the variable region, mutations occur in the variable region. Again, your genome should not be mutating except in the heavy chain and light chain gene locus of B cells. And so this process is known as somatic hypermutation. So you do have mutations going on in your DNA, but these are good mutations. So let's follow through here. So what happens if the DNA gets mutated to these nucleotides? Well, the variable region of the heavy chain and the light chain gene are going to change due to the changes in nucleotide sequence in these codons. So if the repair enzymes ended up switching the C to a T, well, now this codon reads for a leucine amino acid, not a proline. If it puts in an A, well, now the codon reads for a histidine. If it puts in a G, that would read for an arginine. If it puts a C back, by accident or whatever, and it puts in a proline. So we have changed one nucleotide in the variable region, which might change one amino acid in the variable region, which might significantly change the three-dimensional structure in the antigen binding site. So let's see how that's going to play out. So you have these mutations occurring when B cells become activated, and the mutations happen randomly, and you have, remember, an army of B cells that are activating. But not all of them are going to be sent through this entire process completely, because only the ones with the highest affinity will survive this process. So again, let's focus just on that one codon that we chose. We mutated that second cytosine to either C or T or A or G. And if that happens, then that's going to change an amino acid in the variable region of the protein, which will change the antigen binding site. So the first B cell, it's got a proline. The second B cell's got a leucine. The third B cell has a histidine. The fourth B cell has an arginine. So it's changed that original proline into these other three amino acids. That could change the 3D structure of the antigen binding site. So what's going to happen now? Now, that original antigen that was first stimulated this B cell to activate, this antigen is going to be tested again against these antigen binding sites. So initially, there was a proline in there, right? And so clearly that antigen binding site had some affinity for the antigen. So that's great. Well, maybe changing that proline to a histidine, for example, changed the 3D structure so that having a histidine instead of a proline gives you stronger binding, higher affinity. If that's the case, then the cell that's got the histidine would actually go on to the next step in the process. All the other cells here that you see, the one with the proline, leucine, or arginine, those cells would die at this point in the process of B cell activation. So the low affinity versions typically don't make it past affinity maturation process. The high affinity versions do, and that's why we call this process affinity maturation. Mutating random nucleotides in the heavy and light chain will change amino acids, which will change the affinity for the antigen. And so that one here that has higher affinity gets to survive and move on to the next step. So that third B cell, it switched to IgG. They all switched to IgG, but only the third one, with its histidine in that certain position, which has higher affinity for the antigen, that one's going to survive. The rest are going to die. And now what are we left with? We're left with high affinity IgG antibody. So let's summarize here, going back to the beginning. You've got naive B cells that express IgM, IgD on their surface. If they happen to encounter an antigen with their antigen binding site binding to it, that's great. That B cell is going to be activated. It's going to undergo mitosis. You'll have lots of B cells that look like this. Some of those B cells will turn into plasma cells and start unleashing IgM. They will start secreting IgM. No affinity maturation occurred there. So you can make IgM pretty quickly, but you don't undergo that process of affinity maturation and somatic hypermutation and isotype switching. That whole process takes about a week or two to complete. So IgM is made actually within a few days of B cell activation, because all you need to do is switch from making membrane bound to secreting IgM. So that happens. And at the same time, you've got mitosis and some of the daughter cells undergoing these two processes, which we summed up in this video and the last one, isotype switching and affinity maturation. So these happen at the same time. The end result of these two processes is that you will have cells that have switched to making from IgM, IgD to one other type. They could be making IgA, they could be making IgE, they could be making IgE, A, or G, depending on what the need is. So that's isotype switching, and they will mutate. So you're going to have somatic hypermutation and affinity maturation. So then that these cells have better binding. These cells have antibodies that have better binding to the antigen. So now you have higher affinity antibody made by these cells. So that is the process of affinity maturation. And it occurs both in the heavy chain gene locus and the light chain gene locus. So the light chain gene, the light chain protein will also mutate. And you can also get improved affinity that way by mutating both the light chain gene and the heavy chain gene.